Professor Pad is in my room because of internet issue, but now he's connected and he can talk from here.
Yeah, I can see Devesh. Devesh is connected.
Hello.
Hello. Yeah.
Hello.
That is Dr. Devesh Suthar.
Nice to meet you.
Yeah.
COVID has disappeared completely from your country?
Oh, no. I think in Switzerland, they just basically decided the rates are still high. So with the 7-day incidences are still like 1,500 to 2,000. But since the hospital system is not overwhelmed, and there are not so many, it's not overwhelmed. So they decided that because this was always the driving force behind all the restriction is that the hospital system would be overwhelmed and that they cannot provide assistance to even to COVID patients and to the other patients. And this is now not the case. So there's essentially the government doesn't have any people should not be still restricted. They lifted the restrictions, even the mask wearing mandate, except in public transport in Switzerland. So I'm German. So in Germany, this is still, they are more cautious in Germany. So they're to do this way. So how is it in India? How is it in your state?
In India, the number of cases have come down considerably. So now the restrictions are kind of relaxed. The option is some of the respective homes. Otherwise, open areas, it's optional. Some of them wear, some of them don't.
Yeah,
And
so here's also. Yeah, I think this is similar. This is similar here, right? So, I mean, for example, also in the lab or so, we don't wear masks. But then, for example, when I go to a shopping mall or go grocery shopping, I wear a mask because there I'm coming into contact with too many people that I have no control over.
So, yes, true. And what about teaching? Teaching must.
So we went back now to in-person teaching, and there again, there's no strict mask requirement, but there are a number of people of students do still wear masks. So the lecturer doesn't, because anyway, then it's very muffled. So, but the students, some of them wear masks, but most of them don't. And we don't have any. So we want the students to come back. So we don't offer at this point hybrid courses anymore, which we had been doing all along. I mean, some of them had been strict remote, and but then we have also practicals, and they, of course, this was then in a hybrid format, where we basically had then smaller classes so that people could stay further away. But now this is all back to normal. How about for you?
We still have this hybrid system, and also online and offline. Both the things are going on. for the reason that in the hostels they have not lifted the restrictions.
Okay.
So some of the students who are outstation candidates, they don't have a place to stay. Not all PG accommodations are open now. Some of them don't want any paying guest. So they have nowhere to go. Keeping in view their difficulty, we are still continuing online or in hybrid mode.
Yeah, of course, then you have to do this because, but here, yeah, people, yes. I mean, here, most people live actually with their parents. So this most students do. And also the student hostels, they are all open. So everything's open.
Okay. But somewhere one has to make a beginning once again.
Yes. Yeah. No, it's, and then, I mean, the cases are falling. Yeah, and it's about time. People are getting tired of all these restrictions all the time. So this is going to start in what, in 5 minutes, or yes, 45.
Another
right?
8 minutes or so.
Yeah.
How many students? How many students you have in your group?
In total, we are 11.
I see.
That's postdoc, graduate students, and master students, and my technician.
How many postdocs you have working?
4.
Oh, that's a good number.
Yes. Yeah. I try to keep. I mean, a lot of my colleagues, they only want postdocs anymore, but I'm happy to have the entire range. I like to have master students, graduate students, and postdocs. I think this is beneficial for the entire group.
Yeah,
I think the communication is better.
It's a healthy combination because postdocs are adept at many things and they are faster in producing results and they can train the younger graduate students.
So I wouldn't subscribe that the postdocs are necessarily. I mean, a good graduate student after in his second year can actually produce quite a lot on very nice data. So I think it's also the motivation. I think I guess the postdocs at one point when they think they cannot do it in academia, and then it becomes a little bit of a job. So which is not the same motivation to do something. So how about you? How big is your group?
My group has 7 PhD students and 5 masters students.
Oh, so it's also a fairly large group.
Yeah. I think Dean sir has joined. Dean sir has joined. Moment. He was there.
Yeah, he was there. I think he must have seen if things are moving and then.
Yeah. Welcome. Hello, sir. Welcome.
Yes, hello.
Sir,
Sir, you are muted, sir.
Yes, hello.
hello, sir. Hello.
Hello.
Professor
Hello.
Anspang has joined. That is Professor Harish Pad.
Nice to meet you.
Yes.
He is our alumnus and ex-VC of university and a provost right now of one more university.
Oh, wow.
Pushpa,
Yes.
can we invite the dean now to join?
Okay, I'll just.
4:12 it is.
As of now, we don't want anybody else. No, then
Pardon?
before Anne Pang's lecture, we don't want anybody else except us and Harish Pad sir.
No. Inauguration will be there.
No, the VC sir is not required. No, only Dean sir.
Only dean, sir.
So I'd
I'm
ask him
saying
to
dean,
join.
sir.
Yeah, I'd ask him to join.
Yeah.
Okay.
In another 2-3 minutes, he can join you, sir.
Sir is giving welcome address right now.
Okay.
Oh, okay.
So as soon as this welcome address is done, I'll tell him to join.
Okay. Hello. Who has joined just now? Poonam Singh?
Yes, ma'am.
Huh. Okay. Hello, sir. Puran Singh? Dr. Puran Singh?
Oh, yeah. Good afternoon.
Good afternoon. I didn't understand. Okay, sorry. Welcome.
Thank you. How are you?
I'm good. This is the first time we are meeting.
Yeah,
So I
online.
have taken time to find out.
Right. So
Yeah.
I do have few students from your university.
I was a student in CCMB long
Oh,
ago.
when was that?
That was 86. 86
Okay,
to
okay.
88.
Okay.
No, 88. 88 to 90. I
Okay.
was with Dipankar Chatterjee.
Ah. Okay. So that is way before me joining CCMB.
Yeah.
Yeah. So how is situation in your place? Hope it's all well.
It is under control.
Okay, that's good.
Yeah, the numbers have come down considerably, and also the symptoms are mild.
Okay. So
Many people don't even realize that they are infected.
yeah. So you are open for physical classes, or is that still online?
All three are happening.
Okay.
We have online, offline, and mixed.
Okay. All right. Because unlike us, you have a lot of risk because you have students who would like to come.
Yes.
Yeah, in
yes.
research institutes, the population is not that large.
Yeah, but compared to those days when I was there, I think the numbers have gone up considerably. Like
Definitely. I
yeah.
don't know what was the number of PhD students at that time.
That time
Now we have about 150.
yes, that time maybe 20 students and 8 group leaders.
Yeah.
20 to 30 students.
Much more now.
Yeah, right. I think we can start the program.
Sure.
Yeah, okay.
Yes, ma'am.
They are going to play the anthem.
The university song, yeah.
Yeah, university song. Satakshi.
सपना सारग नारा हमें ज्योति जलावी सृष्टि नावरी सरसा सर्जनहारा हमें बड़ोदरा विद्यापीठ सपना सारग नारा हमें ज्योति जलावी सृष्टि नावरी सरसा सर्जनहारा हमें गगन कुसुम कर धरनारा हमें मगन मगन थी फरनारा मगन बाज हमें धरनारा हमें नैयति मिलने हरनारा हमें बड़ोदराना विद्यापीठना सपना सारवनारा हमें ज्योति जलावी सृष्टि नावरी सरसा सर्जनहारा श्री सयाजी विद्यापीठना ज्ञान दीप ने धरनारा श्री सयाजी विद्यापीठना ज्ञान दीप ने धरनारा सत्यं शिवं सुन्दरं मन्त्रनन्दर
Good evening. Respectable Dean, Faculty of Science, Professor Haribhai Kataria, dear esteemed guests, colleagues, friends, and students. I am C. Ratnaprabha, convener of the International Symposium on Proteins and Biology. I feel delighted to welcome you to the symposium. Proteins occupy center stage of biology, being the workhorses of life. There are around 30,000 proteins in a eukaryotic cell to take care of the various activities in which they take part, as enzymes, as defense proteins, as regulatory proteins, etc. Failure in the function of certain proteins results in cancer, neurodegenerative diseases, and metabolic disorders, etc. Most of us are involved in research on proteins directly or indirectly. The symposium is an attempt to bring together scientists working on different aspects of protein research. We have speakers presenting their research on targeted protein degradation, post-translational modification of proteins, error correction in protein biosynthesis, peptide nucleic acid assemblies, proteins in diseases and their degradation, discovery of novel proteins using bioinformatics, and many others. We have speakers delivering lectures as part of the 3-lecture series of the department. Professor C.V. Ramakrishnan lecture series started in 2010. Professor S.D. Telang Memorial Lecture Series initiated in 2009. Professor L.J. Parekh Memorial Lecture Series in 2019. Professor C.V. Ramakrishnan was the founding head of the department. Under his able leadership, the department became major seat of learning and research in biochemistry. Professor L.J. Parekh and Professor S.D. Telang contributed to the growth of the department with their excellent teaching and research. All 3 programs have been initiated by the Department of Biochemistry and the Alumni Association of Biochemistry Department to express our gratitude to the 3 professors of the department for their inimitable and excellent services to biochemistry as well as the department. I once again welcome all of you. I request our Honorable Dean, Professor Haribhai Kataria, to inaugurate the International Symposium and deliver welcome address. At this juncture, I take the opportunity to express my gratitude to Professor Haribhai Kataria for supporting this symposium. And before the inauguration, that I request Dr. Sanjeev Upadhyay to introduce Dean Faculty of Science to the audience.
Looks like Deep Sir is not around.
Just find out with Devish. I think it is worthwhile to mention that Department of Biochemistry is one of the first few departments in the country to teach biochemistry. Actually, there is one more program going on National Science Day. The valedictory function is in progress, and Professor Hari Kataria has to attend both the meetings at the same time. So there's a gap here, and I request all of you to kindly wait for a few minutes before the program is inaugurated. Oh, Sanjeev, maybe you can start introduction and then we wait?
Yes, ma'am. Good evening, everybody.
Mithra, mute yourself. Yeah.
We are very happy to be in this symposium today, and we acknowledge with gratitude the presence of our Dean, Professor Haribai Kataria, a very young and dynamic mathematician who has infused new energy into the Faculty of Science. Sir is always helpful and for organizing any kind of webinars or any events in the faculty. Professor Kataria is an acclaimed professor of mathematics. His research interest primarily lies in the mathematics of fluid flow mechanics. And he is a very astute administrator and has served the university in various positions like provost, syndicate member, student dean, vice dean, and now he is dean for last 4 years. As dean of the faculty, he leaves no stone unturned to improve the academic standards of our faculty. He has introduced training programs to enable master's students crack the NET exams and interacts directly with the PhD students to address any issue they face. He ensures ease of work at faculty, and we are really grateful that he works this way. More than anything else, he is loved by all students and his colleagues because he never says no to most of us who visit him. Over to Dean, sir.
Yeah. Thank you, Sanjeev. Thank you so much. Good afternoon, everybody.
Good afternoon, sir.
Good afternoon. Am I audible, madam?
Yes, sir.
Yeah. Respected Professor Harish Pad, sir, the Honorable Vice Chancellor, Vanita Visram, Women's University, Surat, Professor Ratnaprabha, Head Department of Biochemistry, all invited speakers, colleagues from the faculty, invited guests, and dear participants. Let me take this opportunity to brief you all about our faculty first. Ours is very versatile faculty having 12 departments, 200 teachers and 200 non-teaching staff members, catering needs of 5,000 students of UG, PG and PhD. We have good tradition of regularly updating our syllabi timely and introduce new programs. We also encourage research by signing MOUs with industries and reputed institutes of higher education in India and abroad, patent project writing by publishing research articles in well-reputed journals. Recently, we have signed MOU with Yubat Color Private Limited, a well-reputed industry, and started MSc program in Industrial and Sustainable Technologies from July 2021. See, in this course, the third semester of this program will be benefited, students will be benefited by scholarly inputs from the resource persons of industry. So the industry people are going to teach the complete third semester. That is the part of MOU. And in the fourth semester, students will be having the internship in various industries. I'm glad to share with you that we have signed 16 MOU with different industries, government organizations, and well-reputed institutes of higher education. We have encouraged many of our young colleagues for filing patents, and today we have 11 patents. We have motivated and facilitated colleagues for generating funds from government and non-government funding agencies. In last 5 years, we have mobilized funds of rupees 100 crores. Due to tireless efforts of my colleagues, we have 11 PhD, 22 PG and 87 UG students from various parts of the globe. I take this opportunity to acknowledge the efforts my colleagues for their outstanding performance in research. See, we have 1298 Scopus Index publications in last 5 years, 40,323 citations, 696 I-10 Index publications, 74 books, 125 projects, 44 research collaborations and industrial collaborations. We also encourage our students for their participation in various academic, sports, extension, and cultural activities. Today we are going to complete Science Week celebration under the theme Vignan Sarvatra Pujyate, which is an initiative by Vignan Prasar and Government of India as a part of celebration of 75 years of independence. That is Azadi Ka Amrit Mahotsav. It's matter of great pleasure for me to be part of this inaugural function of an international symposium on proteins and biology. I am happy that Professor Craig Cruz from Yale University, USA, is giving Professor C.V. Ramakrishna lecture on TPD, a new therapeutic modality. Professor Anne Spang from University of Basel, Switzerland, is giving Professor S.D. Telang Memorial Lecture on Principles of Intracellular, and Professor K.N. Ganesh, Director ICER Tirupati, is giving Professor L.J. Parekh Memorial Lecture on Spline Surgery of Peptide Nucleic Acid, updating PNS for multi-standard assembly. Many other eminent professors, including Professor P.V. Balaji from IIT Mumbai, Dr. Rajan, Dr. Puran Singh from CCMB Hyderabad, Dr. Rizwan Khan from Aligarh Muslim University. I am glad that three of our alumni, Dr. Susmita Jha, IIT Jodhpur, Dr. Nilesh Agera, Intas Pharmaceuticals Limited, Ahmedabad, Dr. Verma from Biocon, are also giving their lectures in the subject of current interest. Thus, we have galaxy of eminent professors. I take this opportunity to congratulate the Department of Biochemistry, Professor Ratna Prabha and her team for arranging this wonderful symposium. Congratulations, madam. I take this opportunity and thanks to organizers for inviting me for the opening remarks. I am confident that the participants will be really benefited by expertise of very renowned professors across the globe. And I am happy that Professor Path sir he is present in this inaugural session, and for me, he is a really very wonderful teacher, a very good researcher, and able administrator. सर आप हमें प्रेरणा देते हैं सर. We welcome you, sir, once again. Once again, I welcome all the participants and the speakers. Thanks one and all. Thank you so much.
Thank you, sir. Thank you for inaugurating the program and also for supporting the symposium, sir. Thank you very much.
Thank you. It's my pleasure, madam. Over to Sanjeev.
Not me, he has told it to Bima.
Pushpa?
Ratna Prabha, madam, you can take it forward.
Pushpa, I request you to introduce Professor Harish Path.
Good evening, everybody. It is my pleasure to introduce Professor Harish Path. I will introduce him first as the alumni of our department. He passed out from our department in 1973, so that is nearly 50 years ago. And after passing out from our department, he went on to University of Delhi to complete his PhD. Post his PhD, he worked in several universities in US and established himself as a good biotechnology researcher with biochemistry background, and decided to come back to his motherland. And he came back to our department as professor in 97, and he worked in our department as professor for 4 years. Then the government of Gujarat recognized that here is a person who knows cutting edge research in life science and has got good administrative skills. And he was picked up as director of Perd University, Perd Institute. He went to Perd, he established Perd and got it rolling. And from there, he went to SP University as vice chancellor, which he served for 2 terms, a very, very rare thing to be a VC for 2 terms. And after that, he went to Naipur, Ahmedabad, where again, as a director, he infused a lot of energy and set the institute at to international levels. He has done good work in the area of biotechnology and genetics research. Most of us heard him yesterday and he helped us understand how we can trace the movement of human population through genetic studies. He, for the Department of Biochemistry, he's an alumni member, but is also responsible for us to initiate what we now call the Alumni Association of Biochemistry Department, and he still is the vice president of our organization. He makes sure that we are able to connect with our old alumni and also sees that we collect funds and we meet our requirements in one way or the other. With these few words, I invite Professor Parth to give the keynote address. Welcome, sir. So you're muted.
Okay. Is it coming now?
Yes, sir.
Okay, I'm not seeing the presentation yet. Okay, fellow colleagues and my dear students, very sorry for this technical inability of myself to get into this modern technology era and delay because of this one. I'm very delighted and thankful to the organizers for having me in this meeting. And the symposium on protein biology is very important from several points of view. But the point that I would like to discuss today is the connection of proteins with the genetics. In last 20-25 years, a lot has happened in the genetics or genomics era and its connection to the respective protein structure as well as protein function. And it has come to that extent that even individual genome and the protein structure of that particular individual can also be deciphered with a lot of details if necessary. What I will do is I'll brief you about what we have learned in what we call pharmacogenetics. In other words, what the genetic variance in individual, the way it affects the disease predisposition as well as the drug response in that particular individual. And that whole science of pharmacogenetics has developed in last 25 years or so. And And now what I will do today, and without giving you much information, there's no information in fact, but as a kind of throwing an idea, I would like to extend what we have learned in the genes and proteins which are involved in metabolism. Can we extend the same relationship with the genetic variants and the structure and functions of anatomically important proteins? In other words, structurally important proteins, either the structural proteins themselves or the proteins which makes the structures, anatomy in our system. So my idea of today is to throw open the ideas or the avenues for the young minds to pick up, and then we can see whether we can connect some of our diseases and ailments to the structural defects because of the genetics, as we have seen in the pharmacogenetics. So this is a kind of a loud thinking that I intend to do with you all. Now, human genome, as you know, was done in 2003. And ever since we were wondering whose genome we have ended up with characterizing. And actually, it doesn't matter whose genome we have characterized, because we all are, although we are unrelated, we all are 99.9% same in our genetic context. But the 0.1% difference makes whatever difference we have in inter-individual differences in the health and the disease process. But that 0.1% amounts to more than 3 million variants, or the SNPs and other variants that we have known so far. Let's go back to pharmacogenetics for a while. How did it start? It started by our understanding that most of the drugs have a large populations which do not respond well. We call it non-responders or poor responders to the therapy. And I have listed some of them, like asthma, beta adrenergic agonist. Almost 45 to 70% population do not respond favorably to the medications which are used for this. And likewise, I have listed so many. On an average, 50% of people do not respond to any given therapy for any particular diseases. That's on an average. Now, why is that? And in spite of so many clinical trials, you can see that the number of drugs are even withdrawn after coming into the population use for general use. And that withdrawal also is another spectrum of the extreme drug effect. We call it adverse drug reactions. So on one side, we have people who do not respond to the medications. Other side, we have people, although fortunately small number, who have a severe adverse effect of the drugs. And in between, there is a population which has a good effect. And the withdrawal of the approved drug is because of various reasons. Some of them are listed in this slide. And you can see that these are the ECG-related QTc issues, or some of them have a severe hepatotoxicity or renal toxicity. And so the point we want to make is that we have, the point we want to make is like this, that if there is a protein, in this case it is cytochrome P450, one of the drug metabolizing enzymes, if there is a G in a particular position, it is the wild type. And if this wild type, you will see the drug pharmacokinetics will be something what is shown on the upper graph, upper chart. And you can see the region which is falling in the bracket of what is called therapeutic window. If that one G is replaced by A, and remember we have more than 3 million such SNPs in our genome, in individual genome. And if that A and you can see the tremendous increase in the drug level in the body, because it makes this particular enzymes very slow or sometimes even dysfunctional, and that is why it leads to the accumulation of higher amount of drugs, and that leads to the toxicity of the drug. And on the other way around, if the enzyme becomes super fast, the level of drug doesn't reach the therapeutic window, and those will be the non-responders. And that has been shown in many cases. But most cases, you will see that the genetic variants are related either to pharmacokinetics or pharmacodynamics. Pharmacokinetics is how we handle the drug. In other words, we transport the drug, the drugs bind to the plasma proteins, or the drug is eventually metabolized by a set of enzymes which are responsible. Now, if there is a variations because of individual genetics, those variations affect all of this process. Similarly, the action of drugs, like on receptors binding on ion channels or enzymes. So if the variants are in one of these proteins, then it will affect the function of the drug, the effect of the drug. In other words, there are a number of steps which can affect the ultimate effect of the drug response. And the point I want to make is that most of the genetic variants has been so far related to the metabolism aspect of human biology. Now, this is a very illustrative example that if there is a arginine in the bronchodilator response, in the beta-adrenergic receptors at a particular position, the efficacy of bronchodilator is almost 100%. But if one of that arginine is replaced by glycine, the efficacy falls below 50%. And if both homozygous, if both the arginines are replaced by glycine, you can see the efficacy is insignificant. So this is an example that one nucleotide change can make a lot of difference into the ultimate outcome of the effect of the drug. And this kind of thing, even this is another example where you can see that the drug is omeprazole, which is used for the ulcer treatment. And in either case, if the ulcer is either in gastric ulcer or duodenal ulcer, depending upon your genetic status, if it is a AA, if it is a homozygous for A, you have 100% efficacy. If it is a heterozygous, you have A and B allele, then you have almost 50% efficacy in both the cases. And if it is a homozygous for a fast metabolizers, you can see the efficacy falls below 20%, about 25%. So this is another example that a single nucleotide change can make a complete difference in the outcome of the drug response. Here is another interesting example. The drug is called Xeloda, it's a brand name, and it's used for cancer treatment. The drug is actually a prodrug, and when it is converted to drug by one enzyme called TP, and another enzyme, DPD, converts the active drug into inactive metabolite. So if you take the ratio of these two enzymes, the one which converts prodrug to drug and the other one which converts drug into inactive metabolite, and if you plot the ratio, you can almost see, you can predict the efficiency of the therapeutic outcome. And you can see that if the ratio is less than 1, as shown in the red dots, the effectiveness of the drug is very minimal. And if it is 1 or more than 1, is shown in the green dots, it is a good efficient drug for those people. So what we, this is another example and perhaps more relevant and widely used example where the drug is a Herceptin, which is a monoclonal based therapy for breast cancer. And the drug is very good, but it is very good only in 1/3 of population. And 2/3 are completely ineffective. And remember, this is a breast cancer, so we don't have much time to trial and error way of finding the right drugs. A simple test can predict whether the therapy is going to be effective or not. And that simple test is look for quantitate the number of receptors in the nucleus. If there is a good number of receptors, one can predict that the woman is going to benefit from the therapy. If the receptors are lower number, then it is going to be ineffective, and one can save the time and money, etc., for this particular treatment. So I've just shown you a few examples, but collectively they all tell us that although our medical healthcare system treats all patients as a same individuals, but we have now a way of kind of stratifying the population, that for one given therapy, the population, among population, some of the people may find the drug is toxic and not beneficial. We find the drug is not toxic and beneficial. This is the kind of test that we need to do. Some will find the drug is neither toxic nor beneficial, or the drug is toxic but beneficial. So we have learned how to classify the patient population into one of these 4 categories. And just to give you the global variations that we find, here the circles, the pie chart, the circles are the population which are poor metabolizers for these 2 enzymes, CYP2C9 and 19. And you can see, for example, CYP2C19, that most of the populations are about 20% of them are poor metabolizers for C219. But if you go in Far East, that ratio increases all the way to about 75% in this area on the Far East side of the globe. And the simple point is that the populations are not uniform. Individuals are also, there is inter-individual and inter-population variations in this. And when we develop a drug, we need to keep in mind whether the drug is going to be effective in a particular genotype, or it is going to be effective in all of the populations. And these are some of the FDA-approved biomarkers which are used to test the efficacy and predict the efficacy of the therapies. And here we have listed the enzymes. Warfarin is a very classical example where we have to actually titrate the level of drug, no genetic test. Actually, we have to see how much drug is there in the plasma before we start the therapy. And there are many other examples. So you can see that this list has grown over a period of time. And what we have learned is mostly linked to the metabolism and the functions of protein as it links to the response to drug. Now, see, the genetic variants are there, and they will be there in all the proteins almost uniformly throughout the genome. Now, if there are variants in this set of enzymes which are involved in metabolism, do we have a reason to think that these variants do not occur in the structural proteins? Can we say that my blood vessels and your blood vessels are actually formed exactly in the same way, or there has been some genetic variance between us because of which my blood vessels might be slightly different than yours in terms of size, in terms of diameter, in terms of level of branching? So I want to throw open this new area that we have learned a lot about the metabolism and linked to the individual genetics. But can we also think about the structural proteins and the entire machinery which makes the anatomical structures, so various anatomical structures in our body? Can we think about vasculogenesis, that is the de novo synthesis of new blood vessels during embryogenesis? And whether these proteins, because of their genetic variants, whether they are slightly different, they will not be detrimental, but they will be slightly different. And whether we have reason to think that some of the pathology in human may be because of some of the important variations coming from the anatomical processes, which gives rise to the anatomical structures like blood vessels, like valves, important valves, either in gastric GI tract or in the heart and circulation processes, and whether this valve may be quite similar but not identical in every individual, and slight variations because of the genetics of the individual can also affect the structure and functions of these particular organs, these particular anatomical structures, and whether we should now study the things like angiogenesis, vasculogenesis, and valve formation from this perspective of individual genetic variations. There are also other numerous examples like vocal cord. Now, vocal cord, it identifies the individual with the particular voice and the voice frequency that it emits from the vocal cord. Now, they are all same in many ways, but there may be slight difference because of which my voice is different from your voice. And likewise, there is axonal growth. So n number of anatomical processes. And I just want to leave open this idea that individual genetic variations, as we have linked it to the metabolism and drug metabolism and other aspects of functions, do we have a reason to think that the similar variations, genetic variants are there in the structural components of our body, and whether they relate to the some of the medical or pathological conditions that we see down the line. Just illustration of how the formation of heart during embryogenesis. And you can see that this is the embryo at the 18 days structure, 18 days of time. In about 20 days, there are two endocardial tubes which fuse in a couple of days, and this process goes on 22nd days, 23rd days, and by about 35 days, the heart is formed. So between the 20 days and 35 days, in 2 weeks' time, the heart formation has taken place. And then subsequently, the 4 chambers of heart, as well as the heart valves, have been created. So all this process, needless to say that there are n number of regulators and there are n number of protein machinery which are used for formation of heart and the heart valves. Whether the genetic background of this individual, this particular case, would have some impact on the ultimate functions of ultimate structure and then ultimate functions of this heart is something that I think we should start thinking about. Just to give you an example of the vasculogenesis, there are some regulatory molecules like VEGF, etc., have been identified, and there are many of them. But I'm going beyond that, most of them are transcription factors, but there are a very large machinery of proteins which would be actually forming the structure and functions, anatomical structure and functions that we are discussing today. And all of them, these factors, as well as the machinery which makes this thing, would have a genetic variance, and because of that, they would have structural variations and functional variations because of the structural variations. So you can have a look at the circulation system. You know how complex this the whole structure has developed, and needless to say that genetic variants would be there everywhere, and they would have some impact on the kind of valve and the blood vessels that can be formed, and because of that, some conditions can be attributed to this genetic variations. Now, why should we study it? We should study it because we are already using anti-angiogenic therapies in cancer. Because in the tumor, there are blood vessel formations, and the therapy which prevents that blood vessel and starves the tumor mass of the nutrient is already in use. Then there are, it also represents as the potential new therapeutic targets. If we understand how the genetic variants really work, which are the players, then that offers a therapeutic target for making the process much more safer and better. And the better understanding of more complex vascular dysfunctions like stroke and diabetes have a lot of vascular complications, dysfunction. And if we understand the vascularization, then we have a hope of better management of stroke and diabetes. Genetic development of vascular diseases, and there are also malformation of the capillaries, and there are some conditions because of the malformation of capillaries, there is a uncontrolled bleeding in the brain, and people do die because of this. So these disorders have their basis in genes that also play important role during vascular development, and insights gleaned from studying the developmental roles of these genes that can be a more effective diagnosis and a treatment of these disorders. So in conclusion, the pharmacogenomics, we have learned a lot and has also been applied in the clinic, as I've shown you a number of examples, and has a wider applications where therapeutic window is very narrow, where toxicity window is very large, time of treatment is very critical, and cost of the treatment is very high. So we need to study the inter-individual variation in proteins and enzymes involved in formation and maintenance of clinically important anatomical structures like organs, blood vessels, and valves. The variation in size, shape have important functional relevance in health and diseases. Just to add to this, I was talking to a cardiologist who does the implantation of stent in the blood vessels in India. And he told me that the stents are available in the diameter of 2 millimeter to 4.5 millimeter. In the Western countries, most of the stents which are used are in the diameter of about 3 to 4 millimeter, while in India, most of the stents are used is about 2.5 to 2.75. So there is a kind of a subjective observation of a clinician that blood vessels diameter in India, at least if you take it with all the caveats, are slightly narrower than the other population. And so this gives us a kind of lead to think whether we should study now individual genomics to the structure and the structural component, anatomically important structural components, which affects our health and disease condition. So that is a kind of summary. I don't want to imply that genetics is everything. The genome is largely constant irrespective of cell type and age, but the phenotype varies from one cell type to the next, from 1 year to the next year, and even from moment to moment. And to illustrate this better, you can see that this individual, this butterfly, the 4 states of butterfly have same genome, but their phenotype is very different. And on the right side, you can see this little girl and this famous actress is the same individual, but very different phenotype, the same genotype. And here is a politician, late politician. You can see that the same genotype, but very different phenotype. So I don't mean to say that genetics is everything, but genetics is something. And we should start pointing now from metabolic point of view to the structural point of view. That is the point that I would like to make. These are some of my colleagues who have helped me in my thought process and my actual research. And these are some of those same individuals when they are not in the lab and they are in good mood. This is what they are. And my students who have taught me a lot, so I thank them here. And I thank once again the organizers for having me here. Enjoyed being virtually present in the symposium.
We can have a little quick questions.
Thank you very much, sir, for a very lucid and interesting presentation. Now moving on to Professor S.D. Tellang Memorial Lecture Speaker, Professor Anne Spang. She is a renowned biochemist, cell biologist from Biozentrum, University of Basel, Switzerland. Professor Anne Spang studied chemical engineering at the University of Applied Sciences in Darmstadt and biochemistry at the University of Paris 6, France. She received her PhD in 1996 at the Max Planck Institute. She was then a postdoctoral fellow at the University of California, Berkeley. From 1999 to 2006, she was an independent research group leader at the Friedrich Miescher Laboratory of Max Planck Society in Tübingen. Since 2005, Anne Spang has held the position of Professor of Biochemistry and Cell Biology at the Biozentrum University of Basel. Professor Anne Spang investigates the basics of intracellular transport. She gained an international reputation through her discovery of the maturation process from early to late endosomes. Her research has shown the importance of certain proteins in the uptake of cargo into transport vesicles and the role of certain proteins in mRNA transport and its metabolism. Her research findings are significant in developing the understanding of many diseases which are based on the location of defects in proteins and mRNA in the cell, such as cystic fibrosis, lysosomal storage disorders. She is a recipient of several awards and recognitions like Binder Innovation Prize, elected member of the European Molecular Biology Organization, Walter Fleming Medal of the German Society for Cell Biology, and EMBO Young Investigator. With these few words of introduction, I thank Professor Anne Spang for accepting our invitation, and now I request her to give the lecture.
Thank you very much for this very nice introduction. I feel honored for about the invitation to speak and to present data from our lab. And I hope it's working and you can see my screen now. So thank you very much again for this very kind invitation and deep honor I feel about to give this lecture. So what I would like to do today is I tell you a little bit about what we're doing in the lab, and that is, every day we're questioning ourselves, how is the cell organized? And this is a depiction of a eukaryotic cell. You know that there are many organelles in the cell, and these organelles have to communicate with each other because they fulfill specific functions which are essential for the functioning of the cell, but nevertheless they need some coordination and some talk about it. And of course, we know quite a bit about this communication. So for example, if you think about the secretory pathway or the endocytic pathway, in which basically the organelles along these pathways do communicate with transport vesicles. And I'm going to come back to this in a second, but the way we usually depict actually cells and like in this first scheme is that actually everything is really quite spaced out. And so of course we also do this in pathways that we want to understand, like the secretory pathway where the ER is on the left side and then vesicles go to the Golgi apparatus and then from there to the plasma membrane. However, whether this helps us to understand because we can plainly lay it out, but whether this is really happening in the cell this way is a different question, because the organelles, they just don't swim about in the cell. And the other thing is also for people working interested in signaling pathways, usually what you find is that there's a stimulus, then there are proteins, and then there are positive and negative arrows, and something is going to happen at some point. But in principle, it's kind of sort of assumed that these proteins and the interactions just happen somewhere in the cytoplasm. But if you think about it, how little efficiency that would provide if you want to have some efficient signaling pathways. And so already illustrated a little bit here with the porous signaling pathway, membranes are also involved. And actually in this respect, you could think about membrane as points where you reduce the degree of freedom for the diffusion and actually can concentrate proteins that have to interact with each other and then actually can positively or negatively influence each other to provide a response. So what I would like to highlight is actually that the cell is a crowded place. You have where the organelles are actually stuck against each other, and the question then arises actually, is this just because this is more efficient, that everything is very much compromised and very close together, or is there actually a good reason just that this would perhaps facilitate communication? And so, of course, I would, and I try to convince you and give you an example for this, that we think that this is by design, and this actually that the different organelles are contacting each other helps actually the communication in the cell. But then, if you think about it this way, you also have to think that actually the membranes that are surrounding the organelles, and as well as the plasma membrane, they cannot be equal everywhere. So they need to be compartmentalized themselves. And of course, this is really what we find. For example, at the plasma membrane, you have sites where you put a nutrient transporter or other transporters that help to take up things in the cells. You have sites where you can actually have secretion. You have cell-cell communication devices. You have signal detection devices. If you think about migrating fibroblasts, of course, the front part is differently than the rear part. And of course, also the membrane that is migrating on the substratum is going to be differently organized than the front and the rear. Or if you think about our immune system, cytotoxic T cells, when they actually get ready to engulf the structures, what happens is that they form actually these immunological synapses where they specifically organize these plasma membrane structures and have actin protrusions underneath to provide the membrane to engulf it. Epithelial cells, they have an apical basal and lateral sites. And of course, they have cell-cell junctions, which are specialized domains to provide the barrier function of this epithelium. So, and even in a single cell, like the zygote in C. elegans, the anterior and posterior part are organized in a different way, which actually allows in the first cell division that you get a smaller cell, which is called P1, and a larger cell, which is called the AD cell. And this is actually very important for the development. If they become equally in size, actually this embryo will never develop to an animal. So you need to have compartmentalization on these membranes. And this is, of course, then also translated into the intracellular space, where you have contact sites in which the different organelles can actually communicate with each other. They come in very close contact, and these contact sites are supposed to be sites where you transfer ions and lipids between these organelles. And the endoplasmic reticulum is probably the organelles that contacts everybody, every other organelle in the cell, and therefore could serve as the perfect organelle to sample the cellular environment. And since it's continuous with the nuclear envelope, it actually could directly transmit signals into the nucleus to have a transcriptional response, and therefore making the communication more and more efficient. Besides these organellar contact sites, of course, as I have alluded earlier, we have other types of organization, like the sites where we form transport vesicles. So there you want to get to congregate the cargo which needs to be transported out of an organelle, like in this case, the endoplasmic reticulum, so that you get actually a transport vesicle at a specific site which you need to get to first to concentrate cargo, but also to deform the membrane, to have a bulging out of the transport vesicle that then actually can go on and bud off and go on to the next compartment, which would be the Golgi apparatus. And actually also there, it needs to find the space where it's going to fuse, because it's supposed to fuse with the Golgi apparatus and not, for example, with the plasma membrane or the mitochondria. So you need to get specificity. And so you will have subcompartments of vesicle exit and arrival sites. And all these types of organellar contacts that you can have, or compartmentalization, probably all of them are organized by small GTP binding proteins, which are very often called small GTPases, which is a little bit of a misnomer, because first of all, most of these small GTPases are very, very poor GTPases. And for example, the one I've chosen here, the R1 GTPase, has the propensity or the capacity to hydrolyze GTP with the same efficiency as water. So actually, it is a really very, very poor GTPase, and in conjunction with the GTPase activating protein, the GTP can actually be hydrolyzed. But the function of these small GTP binding protein is actually that once they're activated by a guanylynucleotide exchange factor and then become associated with a membrane, that then they serve as a recruiter of proteins and change the local environment to allow to form a compartment that is going to perform a certain function. And then if this function is no longer needed, then actually the GTPase activating protein stimulates the GTP hydrolysis of the GTP binding protein. And then this GTP hydrolysis renders the protein to become soluble in the cytoplasm again, and renders it into an inactive state. So, but just also to keep in mind, because in the principle we have a rather simplistic view about this and say, okay, a small GTP binding protein is recruited to the membrane, it does their magic, and then it disappears. However, there must be something more about the specificity, which we still don't really understand. And that is just again with the example of this R1 protein, that R1, whose best characterized function is in the formation of transport vesicles at the Golgi, going into the back retrograde direction, it also has a different additional function in vesicular transport to form a different type of transport carriers at the trans-Golgi network to go either to endosomes or to the plasma membrane directly. Likewise, ARF1 has been shown to form vesicles on the early endosome. So therefore, the on and off switch is fine, but the different type of codes in this part that are recruited, or the effector molecules that are recruited, are actually, again, context dependent on where ARF1 protein is activated. And ARF1 does not actually form only transport containers in the cell. We and others have identified roles for ARF1 in mRNA transport, regulation of the cytoskeleton, mitochondrial dynamics, lipid droplet biogenesis, and so on. So there are multiple roles of the ARF1 GTPases, not only in the secretory, in the endocytic pathways, but beyond that as well. So membranes, I guess, and I hope I can convince you so far that actually membranes are compartmentalized, but this also applies to the cytoplasm. And so a number of years now, it has been clear that actually proteins in the cytoplasm can interact with each other to promote phase separation. And this phase separation you can think of as a way as if you would pour oil onto water, and then of course oil and water does not mix normally, and you would get these droplets, these boundaries, and so you have both phases are going to be liquid, but they are not usually not intermixing. And so these phase separated entities, they still, these droplets, they can exchange proteins and RNAs with high efficiency, but they are nevertheless can be rather stable and are separated from the aqueous phase. And they can either be forming by a multivalent protein interactions, or more by disordered proteins together with RNA. And I'm going to come to this later. And the reason we think that you have these kind of phase separated entities is, again, the same thing that you can basically put signaling molecules onto membranes. And that is that on one hand, you can actually make the reaction more efficient. You can use this to organize things. For example, if you think about the centrosome, or sorry about this, or you can sequester constituents of the cell in these phase-separated organelles, and thereby modulate, for example, RNA turnover or signaling. So one of the phase-separated organelles we are interested in are the processing bodies, or P bodies, and in Saccharomyces cerevisiae, these P bodies are the major state where mRNA turnover and storage is occurring. And so, for example, if the cell encounters stress, like starvation or osmotic shock, the first response to stresses mostly is that actually the cell attenuate translation to understand what's going on, and then the mRNA can be put into processing bodies and stored in processing granules, and then actually the cell can figure out what to do, and then either degrade the mRNA or return it into translation. So, but for today's talk, I actually would mostly like to talk about compartmentalization on membranes, and they are in the endocytic pathway. So, by ways of introductions, protein and other constituents very often enter the cells through endocytic vesicles. They will fuse together to form an early endosome. This early endosome is usually covered with the small GTP binding protein RAP5, and this after a while it stops accepting input from the cell surface. We don't know what the signal for that is, but then what it starts doing, it becomes actually a sorting endosome, in which it also changes its shape. It becomes has then tubular components and a more globular part, and from these tubular entities, what we think is basically that proteins are sorted into them to be recycled either directly or through the recycling endosomes back to the plasma membrane or to the Golgi apparatus, while the globular part, there's going to be protein going to deposit into intraluminal vesicles to keep them from signaling to the cytoplasm. And these intraluminal vesicles will then basically stay in there, and this will become a late endosome, which eventually can fuse with the lysosome to form an endolysosome. And then in the lysosome, proteins and lipids will be digested, and then these constituents will be returned to the cytoplasm for the lipids will be transferred for further use. So, but already, if you think about it, if you look at this, the sorting endosome is actually quite interesting because it has this tubular part as well as this globular part, and obviously this is a prime part where compartmentalization occurs, and so this is actually what we like to figure out. So we have this RAP5 positive endosomes that becomes a sorting endosomes where we're not entirely sure which RAP it contains, but the late endosomes, they contain RAP11 endosomes, and recycling endosomes, as important for this talk, they mostly going to be RAP11 positive. So, but the first thing is actually how do you get from the early endosome to the late endosome, and how do you form these kind of compartments? And our way in was actually through the analysis of a mutant in C. elegans, and also through the use of imaging techniques. So what we found is actually that we use the celomocytes in C. elegans, which are cells that are, these are immune cells which have naturally, they're scavenger cells, they have naturally large endosomes. So we could label these endosomes with RAP5 for early endosomes and cherry RAP7 for late endosomes. And what we could, what Dimitri, my postdoc at the time, could show is that actually from a RAP5 positive endosome, the RAP5 would get lost over time, while this same endosome would acquire RAP7. And so at this time, we could then show that basically endosome maturation really occurs, which was before just a concept, but was not definitively shown because the endosomes in mammalian cells are so small, and the imaging techniques at that time were not sufficient enough to actually really observe maturation. It was basically, up to this point, it was just a concept. So we really showed this can really occur. And we furthermore try to understand how this would be regulated. And so in the mutant we had obtained in C. elegans, which was called SAN1, forms the wild-type protein together with another protein, CZ1, forms actually a guanidine nucleotide exchange factor for RAP7, and therefore actively recruits RAP7 onto endosomal membranes, and to turn off the activation of the existing already GTP binding protein RAP5, this complex as well actually displaces the guanine denucleotide exchange factor for RAP5, RAPX5, from the membrane. So you have actually, you provide a switch in which you turn off the activation of RAP5, and at the same time you turn on the activation of RAP7. And this drives then the maturation from an early to a late endosome. However, once we figured this out, and I should say that the data that MON1CZ1 is a RAP7 GEF came from Christian Ungermann's lab in the same year. So, but once you figured out this, then actually the next question comes into mind is then how are different processes are regulated that need to be coordinated from the RAP5 to RAP7 switch, and that is basically the lowering of the pH, the changes of the lipid composition from early to late endosomes, that the proteins need to be recycled, that cargo actually comes in from the Golgi apparatus into the system for proteins that need to be that are required in the lysosome for its function. In mammalian cells, these usually the endosomes move towards the cell center, and you have basically this tubulation and the intraluminal vesicle formation. So a large part of my lab is actually trying to understand how this works. And since these endosomes in mammalian cells, where we also now study this process, are so small, we wanted to come up with a system where we can study this maturation process with enlarged endosomes. So you could either go to super resolution microscopy, but this is not available to everybody. So we tried to think of a cheap and easy method that everybody can use to look at endosomal transport. And so Maria, my postdoc in the lab, what she actually figured out is that we could stress cells for 20 minutes with nigericin, and then wash the nigericin out. And as a natural stress response, endosomes would become enlarged for some period of time. But within 20 hours, the stress was gone, and we also showed that the cells divide normally, and there's no impairment of growth at all, and also no cell death. And just to illustrate the point, if you have basically, and this is just a plain normal wide field microscope, so no fancy imaging techniques require care. So the endosomes are, RAP5 is labeled in red and RAP7 in greens are these little dots. And of course there it would be really difficult to look at maturation processes. But then when we enlarge them, we can actually have this enlarged endosomes, and we can see different domains forming. And then we can also over time, as I can show you in a second, see the maturation processes. And you get basically these large endosomes. So we can follow these endosomes over time with our RAP5 and RAP7 markers or with other markers. And then we can show that basically these endosomes, they acquire RAP5 at a certain point, they lose the RAP5 signal, and at the same time they acquire the RAP7 signal. And actually our signal or our assay works also for later stages. It works very well. And that is that these RAP7 positive endosomes persist for quite some time. And then what we observe is that they stay roundish mostly, but then at one point what we see is that actually they lose their round shape, and then they're becoming smaller, smaller, and smaller until they actually disappear. And so we can then with this assay, we can actually look at endosome formation as well as actually the endolysosomal pathway until you get back to your lysosome. And how these events are regulated is currently under investigation in our lab. But we think that this is a useful assay for a lot of labs who are interested in these kind of questions, because it's really easy to use and doesn't require any super specialized equipment. And we also figured out that you can use different cell types to do this. This was done in HeLa cells, but we also tried neuronal cultures and fibroblasts. One cell type where it doesn't work is immune cells, because there you activate the inflammasome, and that results in cell death. But otherwise, we think it's really widely usable. So now what I would like to discuss with you is actually how recycling works on sorting endosomes, having this compartmentalization in place, and how is this regulated, how does this work? So of course, we are not the first people to start to study this and asking this question. There's quite a bit known about this already. And that is that these tubules that are formed at the endosome are actually formed by, in large part, by sorting nexins. These are bar domain, some of them are bar domain containing proteins, which can actually bind and stabilize tubulated membranes. And so the idea is that basically the sorting nexins would wrap around these membrane tubes, and then cargo would be transported there. And then there are other sorting nexins and other complexes that can bind, and then this would dictate where the cargo that is deposited in these tubes, that it's pinching off, and can go either to recycling to the plasma membrane, the recycling endosome, or actually go to the trans-Golgi network, depending on the timing and the tube formation, how it's formed. However, what has also been observed is actually that there are not only probably tubes forming, but that there are also vesicles forming at the sorting structures. And they would be having a classical code like AP1 or AP3, which would then decide where these vesicles would be going and arriving at the end. Oops, sorry. So our way into this was actually to look at tethering complexes, and that is basically because you do have incoming cargo, you have outgoing things, and if you want to make a vesicle fusion, what you have is basically you have to first capture the vesicles to the membrane, and the long range capturing is done by tethering complexes. So these tethering complexes act all along the secretory and the endocytic pathways. The most well-studied one is, for example, is probably the Hobbs complex, which allows the fusion of Golgi-derived vesicles with the late endosomes, but also promotes the fusion with the lysosome, with the endosome, as well as what is not depicted here, the fusion of the autophagosomes with lysosomes. Its sister is the Corvette complex, which differs only in 2 subunits, but these 2 subunits then promote change the specificity. And while the Hobbs complex fuses membranes that are both Rab7 positive, the Corvette complex fuses membranes that are Rab5 positive. But then there are more complexes around, and for which we don't know, or trafficking pathways around for which we don't know where the function is. And we started out to do a little very simple bioinformatics. This was long before AlphaFold or anything else. Just by sequence homology, we predicted that there might be actually 2 more multi-component complexes in the endosomal system. And we named them Chevy and Ferrari, and they would basically put 2 membranes together, and they could actually then promote the interaction with snare proteins through this VPS33A, B, or VPS45, which is the snare master protein, and then there would be Rab binding domains 41 and 39 for Rab7, 8 and 3 for Corvette. And at that time, when we wrote this review, we didn't know what would be going on with Chevy and Ferrari. So for the Ferrari complex, we made actually quite a bit of progress since we thought about this, and they may actually help to specify domains and subcompartments. So this Ferrari complex that we basically identified has this VPS45, which interacts with snares. It has 2 different subunits which will interact with Rab proteins. The one is Rab11 FIP2, which is binding to Rab11, and then there's Rabinosin 5, which is going to be binding to Rab5. The other thing which is actually rather unusual about this tether is that it's not only does it bind, unlike Hobbs and Corvette, to 2 different Rab proteins, Rab5 and Rab11, but also besides the fusion promoting activity, which is the part which interacts with the snare protein, this is M protein VPS45, it actually contains another protein, RME1 or EHD1 in mammalian cells, and this is actually similar to a dynamine protein, that it actually promotes fission of membrane. So our complex, our tethering complex, has actually 2 activities that need to be in a way coordinated, and that is that on one hand it's fusion promoting, and the other one is fission promoting. So when we started to analyze this complex in C. elegans, and this is now the C. elegans gut. This is the gut lumen with the apical membrane here, and then this is where most of the business usually goes on, and in the Y-type situation. Rab11 would be sitting underneath the apical surface and also in dots throughout the cell. And then when we knock down components of the Ferrari complex, actually we could get a very large Rab11 compartments that are distributed throughout the cell. And I should stress, I'm only going to show you here data from C. elegans, but we find actually the equivalent in mammalian cells as well. And so what we think is actually that Ferrari actually interferes with the recycling to the plasma membrane, because Rab11 is involved in recycling from the sorting compartment to the plasma membrane. To have a better insight in how this is working, we looked at how the Rab11 positive structures would actually act on sorting endosomes. And so this is now where we follow RAP11 in GFP and sorting nexin. The sorting compartment is labeled with sorting nexin that we in red. And when when Yachin, my postdoc, looked at these movies that he made in C. elegans intestine, what you could see is basically that there are RAP11 vesicles coming, staying for a little while, and then moving on throughout the cell. And so then when we looked at this, we thought that actually this comes in the timing is different. So I asked him to measure the timing at the residence time of this RAP11 vesicles on these sorting compartments. And what it turned out is actually that indeed they were different, but if you squish your eyes, you could actually think that they come in rather regularly spaced intervals. Perhaps later that gets a little bit off, of about what it turns out of 7 seconds. And lo and behold, when we actually knock down Ferrari members in this, then actually these residence times, if you look at this, are largely reduced, and you get lower residence times, and also of course then the periodicity is lost. So what we thought, what could be these different intervals, might be different steps of cargo loading into the RAP11 vesicles. And so this, to show the periodicity a little bit better, this is just a different representation. And in black, this is the residence time spin for the wild type situation, where we get basically these 7-second intervals. And if we knock down different Ferrari members, this periodicity is largely lost, and most of the vesicles have only a rather short interaction time with this with the SNCS1 sorting compartment. This is also not only true for RAP11, but also for cargo, which is transported. If when we look at human transferrin receptor or GLUT1, which are both which we both express in the intestine, then again we see these residence times in the wild type that actually now go on for longer times, and we get populations that are really staying on for as long as we can make the movie. And again, if we knock down components of the Ferrari complex, in both cases, this is abolished. So, so these residence times that we get actually when we have a cargo that is overexpressed are getting actually longer, is something I'm going to come back to in a second. So what we think is how a Ferrari works is actually that you have this RAP11 vesicles that comes to the sorting compartment where the Ferrari complex is located. It will dock onto the membrane, it's tethered, and then snare bundle formation occurs, and that leads to fusion pore opening, and then cargo would actually be able to diffuse in, and then the vesicle would try to pinch, and if it can't pinches, then it opens again, and then more cargo can be loaded. And once pinching and fission of the membrane occurs, then actually the vesicle is free to leave. So this is our model, and then the question is, what would be preventing the pinching off in the first round? And so we were thinking about this, and also given that we have this prolonged residence times when we overexpress cargo, Yachin came up with the following model, and that is you would try to form this EHD1, this pinchase would try to polymerize and form a spiral. But then if there would be still cargo, because there's a lot of cargo in this tube, there would be still cargo in the stalk, and then actually pinching would not occur, and this would lead to frustration and depolymerization of the spiral, and the process would begin again. So of course this is something, and this could be compared to the polymerization, depolymerization of microtubules. And there is some credit to this, what we're doing, because the Dynamin takes about 10 seconds to build a filament for pinching. We're here at 7 seconds, and we drew the cargo and the neck size, the stalk to approximately to scale, and you could only fit like a couple of cargos into the stalk of these vesicles. So that would actually kind of fit, but we still have to test the model. So at the end of the day, we think that actually cargo flux might be key into regulating the residence time of a vesicle during kiss and run on the sorting compartment. And so the idea might be that you actually normally you would have to have something which regulates the cargo flow into the vesicle, and just when we overexpress cargo, we would overwhelm the system. And so if this would be the case, we could actually test this in C. elegans relatively straightforwardly, because the cargo that we use is tagged with GFP, and we can do a very, very mild GFP RNAi. So we actually do this by feeding, and we dilute then the GFP feeding RNAi 1,000-fold roughly to get just a sum of the reduction that we can, because we still would be able to need to see the GFP signal, and we can actually quantify the reduction that we get. And when we do that, actually what you can see is that in both cases, for the GLUT1 and for the transferrin, when we do this mild RNAi, we reduce the residence time and we get a higher population of short residence times back. So indicating that this model, there's some credit to it, but we of course need to further explore it. So this is from the cargo side, but then actually we would then predict that there's also something on the sorting tube side, which would be actually regulate the cargo flow. And there we turn to SNCC6, sorting nexin 6, which had been shown previously by Pete Cullen's lab to be involved as a cargo adapter, and we were involved in sorting. So we would predict that if SNCC6 would basically control the flow of the cargo, that if we knock this down by RNAi, or in the main cells we did actually the knockouts, that we actually interfere with the cargo loading, and then again would get actually this clogging and get more rub-11 compartments or longer residence times and more rub-11 compartments sitting on the sorting nexin. And so basically when we knock this out, this is on one hand we see that there's more rub-11 vesicles on the sorting nexin network, and we can show this by increase of co-localization. And as well, if we measure the residence time, we see now if the residence times are actually even under these conditions are long, very, very long. And cargo flux is not very good, because then also these vesicles that dock on the sorting nexin compartment for long times, they are smaller and don't get as big. So we do think that actually cargo and also this cargo adapters do play a role in how cargo is moved through the system, and how actually a rub-11 recycling endosome can be docked on a sorting compartment to take up cargo. So what started out actually initially as a control experiment actually turned out to be not quite a control compartment experiment, because we found that actually the early endosomes, they also can contact the sorting endosomes. And similar to the rub-11 vesicles, or rub-11 endosomes, they undergo kiss and run with this NIX1 compartment. And this periodicity, again, is 7 seconds. And again, this is dependent on the Ferrari complex, because if we knock down the Ferrari complex members, then this kiss and run mechanism is greatly reduced. Okay, so this to sum this up, this part, what we find is actually we have identified this Ferrari complex, which provides kiss and run with of rub-11 and rub-5 positive compartments with the sorting tubules. And we think that, and we have some data that I didn't, for time reasons, didn't include, but we have actually also evidence that there's really cargo flow from the rub-5 compartment into the SNCC1 sorting compartment, and then into the rub-11 compartment. And the idea would be that after endocytosis, you would get actually these rub-5 vesicles can fuse with the sorting compartment and release already cargo, which is required for or destined for recycling. And this would not be binding to the escort complexes, which actually specifically selects cargo for degradation. And then these cargoes would be free to diffuse into the sorting nexin compartment, where they're bound by SNX 6. And this sorting nexin 6 then would regulate the flow into the RAP11 vesicles when they come for kiss and run. And this sorting nexin 6 would bind with a certain affinity, but this affinity of the cargo adapters that are touching the RAP11 recycling compartment, they have higher affinity, and therefore this would prevent the backflow of the cargo, into the sorting nexin compartment. But again, we do still need a little bit of more work to understand it precisely, and but this is all looking very exciting, and I'm super excited about these results. Okay, so now I'm going to switch to the final bit of my story, which was going to be much shorter, and that is I want to talk about compartmentalization of the plasma membrane, and that discussing some recent data we obtained looking at the signaling pathway, the hedgehog signaling pathway, and its receptor patch. So in mammalian cells, or no, not only in mammalian cells, but basically throughout development, it's assumed that actually hedgehog signaling plays an important role, but also in cancer it has been reported to be very important. And the classical view on how hedgehog signaling works is that actually hedgehog binds to its receptor patch, and that leads to the activation and then of smoothen, and then smoothen would inhibit downstream effectors, and that would basically drive then a transcriptional program. In the absence of patch, smoothen would be inhibited, and therefore these other proteins would be active that would inhibit the transcription factor necessary for this transcriptional program. So this is how it works in most metazoans, but then if we look in C. elegans, actually we do have patch. We don't not only have patch, we have also an awful lot of patch-related proteins. However, we don't have, and we also don't have hedgehog, but we have hedgehog-related proteins. There's no smoothen identified. We don't know how patch-related proteins would signal. And then patch, there's one transcription factor, TRA, which is actually for sex specification, but otherwise there is nothing where the transcriptional program would actually add. And so we thought that this would be a super start to look at how this works in C. elegans, and how actually cholesterol, in which these smoothen and patch supposed to be reside, would affect this, how it would work in C. elegans. So the first thing that my graduate student Carla then back did is actually she knocked down patch, which is called patch 3 in C. elegans, and just looked at the worms. And this is just a DIC of the wild type, or it's a phase contrast of the wild type worm. And just by ways of introduction, the worm is C. elegans is very, very simple. It has a cuticle, and in this cuticle, it has two additional tube systems. One is the pharynx connected to the intestine, and the other one is the reproductive tract, which is two gonadal arms, a spermatheca, and then the uterus, and then the exhalate via the vulva. Of course, there are also muscles and neurons, but they are actually not depicted now in this image. But this is basically what you can also really see in the bright field images. Here, this is the gut, and these are basically the gonadal arms with that you can see. Here's the gonad, and here's the uterus. So when we do a knockdown of the patch protein, what immediately is obvious that the worms are much smaller and they are pale. And this paleness is actually indication of the failure to storage fat, which is usually stored in the intestine and gives this black color. And so obviously, when we knock down patch, there's a storage defect of fat in the intestine. And she could also verify this by looking at the lipid droplets and using a specific type of microscopy for which she went to Denmark. She could show that actually patch 3 knockdown worms have less lipid droplets. So around this time, actually it was proposed that patch would be a cholesterol exporter. And so that it would actually take sterols and transport them out. And then once hedgehog would bind, this export of sterols would be inhibited, and actually would then enrich the membrane for cholesterol, and that would actually then indirectly activate smoothen. So the previous model that patch would bind to smoothen, and this would regulate the interaction, the activity of smoothen, was supposed to be wrong, but rather it's actually the amount of cholesterol levels that you have in the membrane. So then we wanted to know whether actually this, because this was just proposed, whether this is actually indeed the case. So Carla went on and actually expressed patch-3 and also mammalian patch in yeast. And then she labeled the yeast with fluorescent cholesterol, and then chased it out and measured in the supernatant how much cholesterol there would be. And then if she does that with a wild type patch-3, then actually she gets a certain efflux of cholesterol. And then if she uses a mutation of patch-3, which would block the cholesterol export, then actually she observed a strong reduction in cholesterol export. Not only could she show this in yeast, but she also showed this in mammalian cells. So she basically had then wild type or patch-3 knockout worms. She labeled them with cholesterol, chased it out, and obviously in the mock you can see that the cholesterol is mostly cleared from the worm, while in the patch-3 animal the gut is still full of cholesterol. So that prompted us to look whether this is more globally, and she so we went on to the mass spectrometry on cholesterol levels, and what we could show that actually in the mock compared to the patch-3 RNAi, what we found is that actually the patch-3 knockdown worms contained more cholesterol than the mock treated counterparts. So where is the cholesterol localized? For this, we used a sensor, a genetic sensor that we expressed in C. elegans in the intestine. And so while there is some cholesterol obviously in the membrane, but it's not so much. What we find is actually there's a quite accumulation of cholesterol at the apical surface that we can actually quantify. So what is the consequence for this? Or if this is really, if we really would assume that we have now here a high cholesterol level, and if that would be a problem for the worm, then actually if we would impair with cholesterol accumulation, we might get actually a rescue of the patch-3 phenotypes. And so if we do a full-blown RNAi, then actually patch-3 never, well, this is lethal, and the worms never reach adulthood. So we twisted it a little bit that we can look through larval development. And so just basically by using plates that do not contain any cholesterol, which we usually have to add to the media because C. elegans is a cholesterol oxotroph, we could actually rescue the developmental phenotype. And moreover, we could improve the fat accumulation in the gut again by just reducing the cholesterol levels. And we can also quantify this by measuring this. So actually, it's the high cholesterol in the cells, in the absence of patch-3, which actually gives the lethal phenotype and the developmental arrest. And so we wanted to see whether, besides in the plasma membrane, this would have the accumulation could occur or would have any consequences. For this, we turned to electron microscopy, and then in the mock-treated samples, you see very nice reticulate ER, lipid droplets, mitochondria, everything, lysosomes, everything seems to be fine. But then when we looked at the worms where we knocked down patch-3, what we observed, we observed these long lines of ER. We essentially almost never observed lipid droplets. And sometimes the mitochondria were also a little bit off. So to understand these ER structures better, I mean, these lines that we see in ER, these lines in 2D with this 17 nanometer thickness are indicative of a sheet structure in 3Ds. And normally you don't see so much in the wild type situation. So to look at this more carefully, what we did is we went to FIPSEM, where you actually mill off, you can basically do a larger volume, and you mill off always after image taking with a laser a bit of the embedded sample, and then image again. And that gives you then a 3D stack. And you can, now we're going through the entire volume, and we artificially labeled for easier, that it's easier to see all the ER membrane in green. And so what you can see, obviously, that the ER morphology seems to be pretty different in the wild type situation compared to the mock. And for simplicity, of course, if you do these kind of stacks, you can make nice movies out of this. And this volume that we basically milled, when we reconstruct this again, I guess you can appreciate that in the mock situation, we have a nicely reticulated ER, while in the patch 3 mutant knockdown, sorry, we have these sheet structures and this really messy looking ER. So this seemed to be that it couldn't be just the cholesterol doing this by itself. So we again looked at the lipidomics and why we didn't see so much changes in the head group composition of the lipids. What we found is actually that the patch 3 RNAi animals, they have shorter and more saturated acyl chain. So there seemed to be a defect in elongation and desaturation, and that usually goes hand in hand. But as a consequence of this is that we think what happens is that actually normally if you have a mixture of, well, the more unsaturated fatty acid you have, the more bendable and the fluid your membrane is. If you have a lot of saturated acyl chains, they become very, very stiff. And they become less bendable. And so what we think is and of course, cholesterol loves these saturated acyl chains and preferentially moves into these areas where you have more saturated acyl chains. And so what we think is happening in the ER case is that actually in these you have lower, well, a lot of saturated fatty acids, and therefore the membrane becomes very stiff, and therefore you cannot build this reticulates so well. But as a second consequence is that because the membranes are so stiff, they may not become bendable enough that you can actually form lipid droplets. And therefore, we lose the fat storage capabilities because you need to form these lipid droplets out of the ER. So that this might be really the case is actually, of course, when you increase the temperature at which the worms grow, then the fluidity of the membrane would increase. And then if we elevate the temperature slightly, then actually do partially rescue the patch 3 phenotype. So we then also went on to look a little bit at the signaling pathway, but I don't have time to really to go into this. But what we think is happening that normally cholesterol is exported out by patch from the cell. And then you have nuclear hormone receptors that actually are important in signaling the fatty acid elongation and desaturation, and this would allow you to form lipid droplets. And of course, with the help of the reticulate ER, where you have a balance of saturated and unsaturated acyl chain, you can easily make this lipid droplets out of the ER membrane. However, when you don't have patch anymore, you accumulate cholesterol, probably not only in the plasma membrane, but also in intracellular membranes. And then you form actually a different nuclear hormone receptor complex, which basically blocks fatty acid elongation and desaturation. And therefore, you get a sheet-like ER, which is very stiff. And this then interferes with lipid droplet formation. And therefore, the worms cannot form, cannot store fat and also not grow efficiently. Okay, So, and of course, we think that mammalian patch 1 is doing the same thing. We know that it's a cholesterol transporter by now, but also this regulation might be very, very similar. So with this, I'm getting to the end of my presentation. I hope I could convince you that membranes are compartmentalized. We developed an easy-to-use assay-to-follow endosomal transport and maturation, and also transport down to the lysosome. We can show that rub11 recycling is regulated by the Ferrari complex that we identified, and that rub5 and rub7 vesicles perform kiss and run on sorting endosomes. And this is probably allowing a robust cargo flow and recycling, and that these processes are dependent on Ferrari. And the cargo flow in these compartments is regulated by cargo availability, adapters, and kiss and run, which is at the moment still a model and we still have to work on. And finally, in the last bit, I told you that that PAT proteins are cholesterol exporters and that they influence membrane stiffness through the transcriptional regulation of fatty acid elongases and desaturators. So with this, I'm at the end, and of course, I have to acknowledge the people who did all the hard work and that are very busy and very motivated to do their work. So the endosomal transport assay was established by Maria Podunovskaya. Ferrari is worked on in C. elegans by Yachin Zollinger and in mammalian cells by Haruno Rashid. And the patch story was developed by Carla Cadena del Castillo in my group. And this is a group pictures from pre-COVID times, and actually when we were snowshoeing. And to celebrate the post-COVID times, we're going to go snowshoeing on Wednesday again. And with this, I thank you for your attention, and I'm happy to take questions.
So now we are open for questions. If anybody has some doubts to clear. This was amazing talk, Dr. Spang. After a very long time, we had such interesting talk. So students who have questions, they can also write on the YouTube page, and I can read it to Professor Spang. In the meanwhile, Professor, I had a question, not actually a question, but I was quite interesting to see that how cholesterol it modifies, and through transcriptional regulation, all through we have learned about the direct effect on the membrane fluidity, but this was also directly affecting the transporter, affecting the transcription and translation of formation of lipids itself. That was very, very interesting for me, being from membrane biology field. I worked on ion channels for my PhD. This part is quite interesting.
Yeah, we were surprised. Actually, normally in my lab, we work on a lot of different things, and I always say we can work on anything, but not on transcription. But then, this is it was for us kind of a bit surprising that we basically that there is this transcriptional control. But it turned out that we, because we were looking then into fatty acid elongases and desaturated, and it became very, very clear that we get differences in expression levels, and then we have to figure out how this is working. And this is, there are these nuclear hormone receptors that apparently then form different complexes depending on what the situation is in the cell and how this needs to be respond.
The other thing I was just, I probably missed, when you do these experiments in C. elegans, when you feed them the fluorescent cholesterol, how do they get into the cells? I mean, are there different transporters that put them into the cells?
Yes, we think that there are different transporters that allows them to get into the cell. So clearly, and it might be actually something, these patch-related, some of the patch-related proteins might actually have the opposite topology, because all these transporters are looking rather similar. And if you look at the NPCs, the Niemann-Pick disease proteins, right, which is also a transporter, but it has actually if you look at the structure, it looks very, very similar to the patch, but it sits in the membrane in the opposite configuration because it pumps cholesterol out of the lysosome, right? So we are actually entertaining the idea that some of the patch-related proteins they may sit in the opposite configuration, and then they would pump actually cholesterol into the cell and not out.
So may I ask few questions, Dr. Spang?
Yeah, please go ahead, sir.
Yeah, so fantastic talk, Dr. Spang. Beautiful work. So I have few general questions. One is Ferrari specific for the lysosomal transport vesicles, or does it also have role in other vesicle transport, for example, to the plasma membrane or to other parts of the membranes?
So we think it's actually specific for recycling to the plasma membrane. So what we see is that actually these RAP11 vesicles, they are destined to go to the plasma membrane. They can actually stop on the way also on different sorting compartments and take up more cargo, but they're going back to the plasma membrane. More importantly, what we also found, but I didn't want to talk about it too much because it's getting too confusing then. So this would be going back in polarized cells to the apical surface. But then we also found that RAP10, which would be recycling to the basolateral side, is also doing kiss and run in a Ferrari-dependent manner. So the two main recycling pathways would actually be dependent on Ferrari. However, I have to stress that we think that this acts on top of the classical recycling systems that many people have worked on. So we just think that we basically discovered an additional pathways using the same, that makes the system more robust than just, and that is a little bit like a distillation process. So you don't have to transport everything that you want to recycle quickly all the way down into the cell, but basically you can have already a way early on to bring back things to the plasma membrane that you actually, like transferrin receptor, which you just want to have iron loading. For the rest, you don't care so much.
So my second question is related to your second part of your story, that is PTCH, if I remember. Oh, so what you saw is when you did knockdown, there was a significant difference in the number of lipid droplets. Since lipid droplets contain various kinds of lipids, but it seems based on your data that this is specific for cholesterol. So do you also see a difference in the accumulation or turnover of other lipids in addition to cholesterol?
Oh, so the thing is, like, actually we see an accumulation of cholesterol, which would basically, in principle, tell you that you get more lipid droplets, right?
Right.
But actually, this cholesterol sits more into, we think, intracellular membranes. And the other type of lipid droplets that you get is actually triglycerides, right? Okay. So it's more, we think, as a consequence. So you still might have triglycerides, but they cannot, because the membrane, the ER membrane is so stiff, because the bilayer to make the lipid droplet, you create a lens of neutral lipids that then expands and then essentially butts out of the ER bilayer, and the lipid droplet itself is only going to have a monolayer around it of lipids. So we think that we have so much cholesterol and this short saturated fat acyl chains that actually the membrane becomes so stiff that you actually cannot bulge it out and form these lipid droplets. So it's really a mechanical constraint. Okay. We did not find so big. Well, we found some difference in the TAC levels, but they were not so. It is not that they don't have any neutral lipids, but they cannot put them in the lipid droplets, and then they cannot metabolize them. They're stuck basically in the ER.
Thank you very much, Professor Spang, for a very interesting lecture. If there are any more questions,
for having me.
If there are any more questions, I request the audience to write to her and get the answers from her. Now we move to the next speaker. I am delighted to introduce today's speaker of Professor C.V. Ramakrishnan Lecture Series, Professor Craig Cruz. He is the J.C. Malone Professor of Molecular, Cellular and Developmental Biology and also holds joint appointments in the Department of Chemistry and Pharmacology at Yale University. Professor Cruz is the Executive Director of Yale Center for Molecular Discovery. He received his bachelor's degree in chemistry from the University of Virginia in 1986. He performed research at the University of Tübingen as a DAD fellow. As a graduate student at Harvard University, he cloned and purified MEK1, that is MAP kinase, which controls cell growth. He subsequently joined Yale University as faculty in 1995. His research interests focus on chemical biology and controlled protein degradation. Cruz is a pioneer in the field of targeted protein degradation, and he uses it for the pharmacological modulation of protein levels. He studied the mode of action of epoxymycin to start with and found that it is a potent and selective inhibitor of proteasomes. Later on, he developed the FDA-approved anti-cancer drug, carfilzomib, also known as Kyprolis, used in patients with multiple myeloma. He developed proteolysis targeting chimeras or PROTACs. He is the founder of Arvinas, the first biotechnology company to bring PROTAC drugs into clinical trials. In 2019, he was named an American Cancer Society research professor at Yale University. Arvinas uses the PROTAC technology to treat cancer, neurodegenerative diseases, and others. Two orally bioavailable PROTACs targeting the androgen receptor and the estrogen receptor are in clinical trials. He is a recipient of several awards and honorary doctorate. He received Swedish Pharmaceutical Society Schiele Award, Boehringer Ingelheim Foundation Heinrich Weiland Prize, American Cancer Society Research Professorship, Pharmacia Aspect Award for Experimental Therapeutics, Royal Society of Chemistry Khorana Prize. These are only a few of the awards and recognitions he has received. It's a very long list. To name a few from the long list, I have chosen these. With this brief introduction, I thank Professor Cruz for accepting our invitation, and I request him to deliver the lecture.
I'm having difficulty sharing my screen. So if
you can see the following things in the bar below. That is a mic, a camera, then the one for captions. After that, you see an arrow pointing upward. Click on that.
Yes. I see that. And when I share, I think the problem is on my side. So forgive me, I'm afraid I'm going to have to restart Google. And so if you can give me about 30 seconds to refresh my computer.
Yeah, sure.
Excuse me.
It's not a problem. Yeah.
Can you hear me?
Yes, yes, we can hear you.
Thank you so much. Thank you for that very kind introduction, and thank you for the invitation to present our work today. It's a real honor to be giving the Ramakrishnan a lecture, and I'm very happy to be sharing our story with you. And so what I want to do today is tell you about my lab's interest in developing new technology. And I don't think I need to explain to this audience the importance of developing new tools, new approaches, and the impact that new technology development can have on both research, diagnostics, but also therapeutics. And I just illustrate 3 examples here that have really transformed how we do research as well as new therapeutic approaches. The problem I want to address is one that's been around now for 20 years. Ever since the genome was decoded, we know all possible drug targets. Unfortunately, only a fraction of those 20,000 potential drug targets are actually currently druggable in the sense that they have an active site or a small molecule binding site. Now, the biologists and the physicians have long told us what proteins are relevant for disease, but that overlaps with the druggable space fairly limited and leaves a significant amount of these drug targets as currently unaccessible or not pharmaceutically vulnerable. And so what I would like to do today is make a case for a new type of pharmacology, one that's not driven on the current focus on active site inhibition. And so if I can just state the obvious, we currently live in a paradigm whereby small molecules bind to pockets on enzymes, and by binding those pockets, and sometimes you might need to bind 98, 99% of a population, you get the clinical benefit. But because most drugs are reversible binders, when they fall off, you lose the clinical benefit, requiring excess number of molecules to ensure that if one molecule falls off, there'll be others to take its place. And this excess of drug is both challenging to achieve and maintain, but it also sometimes creates problems in terms of off-target issues and side effects. So I'd like to describe a different approach, one that's not based on occupancy, but one that is based on an event. And the event is that a small molecule can be used as a recruiting element. It does not have to have any biological activity itself, but it can recruit part of the machinery that's normally involved in protein recycling to now recognize that protein as a substrate for the ubiquitin proteasome system and eliminate it in a catalytic way. And this is a technology that is a hetero-bifunctional approach that we call PROTACs, or proteolysis targeting chimera. And it's a technology that I first developed in collaboration with Ray Deshays, and we published now over 20 years ago. The technology, as I said, is a relatively simple one in the sense that there are 2 ligands. One ligand binds the protein we want eliminated. The other end of the molecule binds to a class of proteins called E3 ubiquitin ligases. And that this heterobifunctional molecule works as a dimerizer. It brings the protein of interest into close proximity to the E3 ligase. And even though the protein of interest is not a natural substance, the induced proximity is sufficient to now tag this protein with ubiquitin and leading it to be destroyed by the proteasome. And so it's this ternary complex that is absolutely critical for the activity of this class of molecules. Now, a key part of this is that the PROTAC is not destroyed and is able to undergo multiple rounds of recruitment tagging, and therefore it works in a catalytic manner, and therefore can also work in a sub-stoichiometric manner, and therefore you do not need excess drug. You can actually use less than 1-to-1 to be able to achieve the same clinical benefit. But once tagged, as I said, the proteasome recognizes the polyubiquitinated protein, cleaves off the ubiquitin, and that gets reused. But the protein is then degraded into short peptides that are further recycled into individual amino acids. So the key part of this is that we're hijacking a natural process that is part of cellular protein homeostasis, but we're doing it in a way that is has clinical benefit. Now, we initially started doing this with a chemical biology approach using a small peptide that could serve on one end of this PROTAC to engage the E3 ligase. And in a series of proof of concept papers, we demonstrated that this concept works using molecules that target the androgen receptor, the estrogen receptor, and a variety of other improvements to the technology. But it was clear that this was not going to be a therapeutic approach. And that's because of the peptide. It was not did not have the desired pharmaceutical properties. And so in 2009, I set out to come up with a small molecule to replace this peptide that was engaging the E3 ligase von Hippel-Lindau, or VHL, with a small molecule. And I should say, I meant a team of researchers on the chemistry led by a very good graduate student, Dennis Buckley, and a postdoc, Alessio Ciulli, who is a structural biologist. And together, after synthesizing over 280 molecules and solving co-crystals of 14 molecules bound to VHL, we were able to come up with a small molecule that bound to the same site as the VHL ligand. And this now allowed us to think about a all small molecule version of our PROTAC technology. So we call this PROTACs 2.0. And so what I'd like to do for the rest of my talk is just describe some of the advantages that I think are inherent in this technology, using examples of various PROTACs that we've generated over the years. So just first of all, I want to describe some of the key concepts in terms of designing. We call this linkerology. This is simply medicinal chemistry. We find that when you have two ligands, how you design the linker in between can have great impact on how the protein is degraded. And some of the considerations that we take into this design are the length, the composition, as well as the rigidity of the linker to be able to afford potent molecules. Now, I want to just give you an example of this, a fairly recent PROTAC, a receptor tyrosine kinase targeting PROTAC. In this case, we started with a known ligand to a tyrosine kinase. This is lapatinib. It binds to EGFR, as well as the EGFR homolog HER2. In fact, people argue that it's one of the most selective E3 ligases that are, most selective tyrosine kinase inhibitors are out there. Now we've converted this to both a PROTAC that is active as well as an inactive control. By inverting the stereocenter here, we're able to inactivate or prevent the E3 ligase VHL from binding. And this is a very important inactive control because it has the same physical chemical properties as the active PROTAC, but being unable to engage the E3 ligase, it can only work as an inhibitor. And so by comparing an active degrader with an inhibitor that has the same properties, we can get a better understanding of what can be gained by degrading something versus inhibiting it. And so what you're looking at here is epidermal growth factor receptor that's being degraded in a dose-dependent manner with this lapatinib-based PROTAC, but at the diastereomer that is unable to engage VHL over the same concentration range does not degrade. But what I really wanted to show you was this concept of linkerology and linker length in particular. Here again, I've shown you EGFR, but now I'm also showing you the other protein that is engaged by lapatinib, HER2. Both of these proteins are degraded when we have a linker length of 2 polyethylene glycol, or PEG units. However, if we increase the linker length from 2 to 3 PEG units, we can retain EGFR degradation, but we have lost the ability to degrade HER2 simply by changing the linker length. And we've seen these types of results with other targets, other ligands, other PROTACs. And so it really gives the medicinal chemists a new tool for designing and specificity. And so it's kind of ironic that we've taken the most potent, the most selective inhibitor, and we've turned it into an even more selective degrader. Now, this next example is one that's targeting a different E3 ligase, cereblon. And I need to just give you a little bit of background on cereblon. Cereblon is the target of what's known as molecular glues. They work very similarly to PROTACs. They have an unfortunate history in the sense that thalidomide, which is kind of the canonical molecular glue, was given to pregnant women in the 60s to treat morning sickness, and was shown to be a teratogen that affected the limb development in the resulting fetuses. It was known basically 10 years after we started PROTACs, it was identified that these worked via targeted protein degradation. And they target a transcription factor important for limb development. But they also target other proteins as well. And I just want to show you, here is cereblon, here is this imide, thalidomide. It binds a neo-substrate, and by bringing this neo-substrate into this complex, they can now get ubiquitinated just as a PROTAC. But whereas PROTACs are two ligands with a linker, this is much smaller, as you can see, and is very difficult to design proactively a molecular glue that goes out, that will degrade a particular target of interest. Now, there are variations on thalidomide. We now know that they can bind and degrade additional proteins. These are two proteins that are transcription factors involved and required for multiple myeloma. And pomalidomide and lenalidomide are successful FDA-approved drugs for these diseases. We know how these proteins engage, how these neosubstrates are recruited to the cereblon. It's hard to see. Here's pomalidomide in here, but they really work by inducing a protein-protein interface, a PPI, by like a molecular glue, by bringing these proteins together. This shows just another example of this. There's a loop here that is engaged by one part of the imide in the context of the cereblon face, and that this together makes a ligand that now will bring these two proteins together. This is the transcription factor important for multiple myeloma. This is the transcription factor that is involved in limb development. And as you can see, there's a nice structural overlay that is responsible for this recognition. I should say that the structure was solved by a former undergraduate of mine at Yale, who's gone on to do some really nice things at Celgene, and now is at the new venture called Neomorph. Now, as I mentioned, these glues do differ in the sense that they are small. They bring these two proteins together, and they induce this new PPI. The advantage, however, of a PROTAC is that one can separate the recognition elements into two ligands, one for protein B and one for protein A. And then through that linkerology I described, one can get to the same place, that is an extensive protein-protein interaction, but you can do it in a more thoughtful, rational way. And so even though I think molecular glues are very attractive from the point of view of having a small molecular weight, it's really hard to think about targeting a particular protein with this approach. Whereas the PROTACs are larger, but they do have the ability to go after a particular target. Now, what I want to describe is a PROTAC that still uses a imide, but doesn't use it in the context of a molecular glue. We are just using these molecules now as one end of a bifunctional molecule that's going to engage cereblon this time. So I don't want to confuse you by thinking that this is a molecular glue. Is just the traditional PROTAC. So I want to talk about going after a different target. This case, we're interested in a epigenetic reader. BRD4 binds to acetylated lysines and is involved in turning on the transcription of a undruggable oncoprotein called MYC. And because MYC is not currently druggable, there's been a lot of interest in coming up with small molecules that will block the function of BRD4 to control MYC levels. And there are many different companies that have developed these inhibitors. The problem is with the biology of BRD4. BRD4 function is so critical for life that nature has evolved mechanisms to sense any loss of function and to compensate by now turning on the expression of BRD4. So it's hard to see here, but there is pre-existing levels of BRD4 in this lane. As we treat with increasing amounts of the inhibitor, we're now inducing high amounts of BRD4. Now, what that means for therapeutic intervention is problematic, right? If you have a pre-existing amount of BRD4 and you have an inhibitor, but the inhibitor induces more BRD4, you now need more inhibitor to now get that maximum saturation. So you increase BRD4 inhibitor, that increases more protein. So you're in this pharmacological arms race, which I would argue you'll lose because ultimately the cell will just simply make more and more BRD4. You'll lose that maximum occupancy. You'll lose the degradation. You'll lose the inhibition. Now, what we've done is we've taken this inhibitor and coupled it to an E3 ligase recruiting element to make a PROTAC that works very, very potently. It's a picomolar PROTAC. In fact, we can routinely now get picomolar degraders, and that's because of the catalytic multiple recruitment mechanism that we're seeing here. Now, I've shown you this before. This is the induction of BRD4 when we have the inhibitor, but I want to show the downstream, the true target of this approach, MYC, is really not affected because of the escape mechanism here. So the inhibitor is not working. However, if you make a PROTAC, we can degrade not only what is already existing here, but we can degrade whatever the cell is trying to make in terms of BRD4 function. And that now results in a more profound inhibition of MYC, and we're now beginning to see cell death. And so this is what a concept that we call differential biology. What do you gain by degrading a target that you don't get by inhibiting a target? So here is an inhibitor. We get one function if it has just simply a hydroxyl here. We get a different outcome if it's a linker and an E3 ligase recruiting element if we turn this into a PROTAC. And so we're always thinking about where is the best place to deploy this new technology that we've developed. The next question that I want to discuss is this concept of how selective do these recruiting elements have to be? Now, I already showed you lapatinib and how we made it more selective, but that's a little odd because it was just only hitting, as an inhibitor, only hitting two different receptor tyrosine kinases, EGFR and HER2. We wanted to purposely make this harder by choosing a much more promiscuous ligand, and feritinib will bind to over 130 kinases. And we just wanted to ask if we now convert feritinib into a VHL-based PROTAC or a cereblon-based PROTAC, would they still bind to all of these kinases? And more importantly, would we actually get degradation of all 133 different kinases? And so the way we did this is known as a kinome scan. Each of these bars, these gray bars here, represents a kinase that is being inhibited by the warhead feritinib. Each black bar represents a kinase that's bound and by VHL-based PROTAC. And as you can see, there are some kinases here at the top, the black, that are equally inhibited by both the inhibitor as well as the PROTAC. But you'll see that there are some kinases that are no longer able to be inhibited by the PROTAC. Simply by adding a linker and a recruiting element, we change the specificity of this recruiting element. Now that's true for VHL. It's also true for cereblon-based PROTACs. Again, some kinases here, as you can see, have the same engagement as the warhead, but we also see some kinases that lose that type of engagement. Now, interestingly, there is not a direct overlap between the VHL cereblon-based protects, despite the fact that the attachment point and the linkers are the same. Depending on what you have here, will dictate what binds here. Now, that's just binary interaction. What we really want to know is, are we getting degradation? So we actually did 2 experiments, the PROTAC that's engaging cereblon, what proteins are being degraded, looking at kinases degraded when we add VHL-based PROTAC. And I want to just focus that on the majority of the kinases known to be bound and inhibited by the warhead ferritinib are not being degraded by either one of these. So we've gained specificity of degradation, but more curiously, we found some that were only being degraded by VHL. We found some that were only being degraded by the cereblon-based PROTAC. So we were wondering, since there were differences in the binary interaction, could that possibly explain things? So we started looking at this a little bit more carefully. Does the affinity of the PROTAC for kinase correlate with the ability of that PROTAC to degrade it? So we replotted the data. In this case, we were looking at the ability to bind a particular kinase and the ability to now degrade a particular kinase. So if you see down here, there are examples of things that are high affinity and great degradation, and that makes sense. You imagine if you bind well, you degrade well. But it got more curious because we did find some examples that of kinases that were not being degraded, but we knew were being still bound by the PROTAC. And conversely, we knew things that were not being bound very well, but was still being degraded. In particular, this p38 alpha binds the PROTAC with only micromolar affinity, but was being degraded at the in the nanomolar range. And so that was quite interesting, and it got us thinking about what is important for degradation. It's clearly not binding, the binary affinity of a PROTAC and the substrate. So if it's not the binary, could it maybe be the ternary, the protein, and the dimer in between? So to test that, we took beads in which we had mobilized our E3 ligase and added our dimerizer or PROTAC and just went fishing for kinases from a lysate. So this has all these different kinases in it. What could we pull down? And we found that we could pull down CMET, had nice affinity, binary affinity, and it also, we knew previously that it could degrade. So again, that made sense. Ternary complex correlates with degradation. We knew that SLK had similar binary interaction, was not being degraded, and that was consistent with its inability to form a ternary complex. And P38 had low affinity, binary, but still could be degraded, and we can see that it was due to the fact that it could form this ternary complex. And so what we think is happening is that if you have a weak interaction between the recruiting element of a PROTAC and the target protein, that weak interaction can be compensated by stabilizing protein-protein interactions by the overall protein interaction face. And so that's quite exciting, and it's changed how we think about designing. We now are making molecular glues. We're not focusing solely on the individual affinities of the ligands. We're looking at the overall ternary complex affinity. Now, this next idea comes from what I began with, and that is occupancy-driven pharmacology versus event-mediated pharmacology. And this is a story that deals with prostate cancer, which is driven by the androgen receptor. The androgen receptor is a transcription factor that will bind testosterone, and drugs have been developed that will now block testosterone binding. Enzalutamide is state-of-the-art molecule that will bind and prevent testosterone from turning on the androgen receptor. We've turned this now into a PROTAC. We can very nicely degrade androgen receptor with a maximum degradation of 90%. We haven't optimized this in this experiment, so we're seeing single-digit nanomolar. Now, how does this degrader compare to inhibitor? So in blue is the inhibitor, enzalutamide. This is a dose response curve. You can see that the drug will block the growth in cell culture of this human prostate line, which is not surprising. You turn enzalutamide, however, into a PROTAC, and you can see in red, we can come up with a much more potent anti-proliferative molecule. But I would argue that's not a fair comparison, because the inhibitor is a product of hundreds, if not thousands, of hours of medicinal chemistry optimization. I think a better comparison would be the very large degrader compared to the inactive degrader that still has an inhibitor end on it. And so the delta between red and green, that difference is what you gain by degrading something versus inhibiting something. But I wanted to talk about this concept of target engagement. And now we're able to study this in this particular system because prostate tumor cells, some prostate tumor cells, have acquired the ability to escape the action of this drug by making their own testosterone. So let me say that again. It's kind of crazy. The tumor makes testosterone, so it can make a competitor for the drug. And so instead of being the drug binding 99% of the time, when you have more and more testosterone being made, you drop from 99 to 80 to 70, 40, and you lose the therapeutic activity of the drug. We can recapitulate this phenomenon in cell culture using an artificial androgen. In this case, the biology we're studying is just the ability of enzalutamide to kill cells. At low competition, you can see in blue, we can kill cells very nicely. But as we increase, the competitor, we're keeping enzalutamide constant, but we're increasing the competition, we lose that biological activity. And I've shaded here in blue, this is the concentrations of testosterone that are found in tumors, clinical samples, biopsies from patients that have failed enzalutamide. So these are physiological anti-tumor concentrations, anti-enzalutamide concentrations of testosterone. However, if we take enzalutamide, make it into a PROTAC, that PROTAC can still have the same biological activity, even at high concentrations of the inhibitor, because transient interaction leads to tagging and permanent destruction. And moreover, in a catalytic way, it works over and over and over again. So this is important because we don't need that maximum saturation of the drug. And this, I think, is really important because it uncouples the traditional pharmacokinetics, PK, from the traditional pharmacodynamics, PD. Normally, to get the clinical benefit, PD, you need large amounts of drug, PK, but that's not the case for PROTACs. So I hope I've been able to show you that this is a new potential therapeutic. What I've done in this slide here is just simply list the characteristics of what I would argue are some of the advantages or characteristics of a perfect drug, if you will. And I wanted to show that PROTACs ticks all of these. I want to highlight a few. Scaffolding functions are not currently addressed by small molecules, as well as the ability to work in a catalytic mechanism of action. All of these are now possible with PROTACs. And it was mentioned in the introduction, a company I started, Arvinis, has taken this concept from my lab now all the way into the clinic with 2 orally available drugs that are targeting androgen receptor for prostate and estrogen receptor for breast. Both of these are in phase 2 clinical trials. Both of these have shown to work in terms of degrading the target protein. Taking biopsies from patients treated with these, they can actually see the levels of the protein go down. So these are the first two of what's going to be, I think, a very crowded space as more and more PROTACs enter the clinical trials. I hope that somebody will be successful and that this will help patients in a series of different diseases. So with that, I just want to acknowledge a very talented team of biologists and chemists that have made all of this possible. I'm going to stop there so that we have some time for questions. And I just want to thank you again for your attention and for the honor of this lecture. Thank you.
Questions from the audience?
Professor Craig, if I can ask, is the technology itself patented, or is it open for all to try, this Prozac?
So there are patent applications that have not been ruled yet. There has not yet been a patent on the entire technology. Maybe to answer the question differently, we started Arvinas, or I started Arvinas in 2013. Since then, there have been numerous companies that have been founded on the concept of targeted protein degradation, and every large pharmaceutical company has an effort, either internal or external, on PROTACs. And so they seem to think that there's a lot of space that's available for additional innovation in this area.
Okay. Thank you.
Can I ask?
Please.
These molecules are purely synthetic.
Yes.
So the toxicity levels have been checked, or they need to be checked?
So they've been tested in preclinical models, and now they've been tested in humans. Excuse me. So the phase 1 trials have determined that the compounds are safe and tolerated, and the adverse events that have been observed with the prostate and the breast cancer are not the severe grade 3 or 4. They are the grades 1 and 2, and moreover, they do not overlap, which suggests that there is not a technology-wide toxicity problem. There are target-specific toxicities, just as you would imagine with any drug, but there is nothing that will limit, I believe, this technology from being applied across a lot of different indications.
I wonder whether the same PROTAC, the backbone of the PROTAC, can be kept same, and the two binding ends can be changed.
Yeah, so we certainly can swap out different recruiting elements. However, because we're looking for an induced protein-protein interaction, that linkerology, the linker length, the composition, the rigidity, all of that has to be optimized for each pair of proteins we're bringing together. But that being said, we have made over 25,000 PROTACs, and when you're dealing with well-characterized ligands, that's 25,000 linkers. And so we really have a good idea of how to bring proteins together to get the maximum interaction.
But to develop 25,000 PROTACs, how much time it would take?
Well, I've been working on this for 21 years. Arvinas has been working on this for the last 8 years.
So, I'm just wondering about the technology, that at one point of time, how many different PROTACs can be evolved or designed?
Well, it's relatively straightforward to make a PROTAC that works in a cell culture, right? So taking two ligands and now adding a linker. A chemist in the make 25 PROTACs, and that most likely one will work, and it would be very useful for a reagent for research. The reason why so many PROTACs have been made is due to all of the pharmaceutical properties that are required for a successful drug candidate, and so that was the effort of Arvinas.
Can I ask, like, is it automated or how?
It is not automated.
It's not automated.
No, it's medicinal chemistry.
Okay, that's great. Yeah. Any other questions?
I have a question for Dr. Craig. Dr. Craig, thank you very much for a nice talk. So yeah, I think one of the or kind of initial bottleneck could be to design new PROTACs for different other organisms other than mammalian cells. I think I don't know this area that well, but I believe most of the PROTACs which are available are majorly targeting the E3 ligases and their substrates in mammalian cells, I believe.
Correct.
Oh, so let's say if someone wants to use those PROTACs for infectious disease pathogens, so it may not be that easy to design PROTACs, but if the existing PROTACs are available as library, commercially available libraries, I think it might be possible to screen them and let's see what work in a pathogen. So I wanted to know if those PROTACs are available from your lab or from companies as libraries.
They're not available for as a library, but I would argue that may not be successful because of the unique individual protein-protein interaction that's necessary for a successful PROTAC development. It's unlikely that a random pathogen protein that you want to degrade is going to work in the context of something that's been optimized. And so that's why instead you should just simply develop a PROTAC specifically for that pathogenic protein. And that's being done. The one caveat I will say is that there has to be an E3 ligase within that system. So for example, prokaryotic targets cannot be, intracellular targets cannot be degraded. So other than that, it certainly is possible if yeast, plants, parasitic malaria, if it has an E3 ligase and ubiquitin, it should be working.
Yeah.
I will say that Arvinas has started a subsidiary to develop PROTACs for agriculture. Okay. And the idea is instead of going after particular proteins using a GMO approach, if one can just simply knock down proteins that are involved in pathogens or have other types of desired properties.
Thank you very much.
You're welcome.
So one little question I had in mind. Can we use this technology to lead a bacterial cell to a macrophage, say, for example, for quick grabbing so that they can tag like an antibody, pull them both together?
So this technology is specific to E3 ligases, but in general, the concept of a hetero-bifunctional dimerizer has been validated, right? And so this is, I believe, the first example of a chemical biology concept that's gone all the way from concept into the human and been validated. And so this idea of dimerizers has gotten the attention of pharmaceutical companies. So they're starting to think about this in different ways. And so my colleague, David Spiegel, has come up with something called ARMS, antibody recruiting modalities and molecules. And so these are heterobifunctional molecules that will bind, say, to the surface of a bacterium, and now will recruit the immune system to recognize that. And so I think that an ARM would be an equivalent, if you will, of a PROTAC for the purposes that you want.
Do you see
Thank
a
you for the
possibility for treating autoimmune diseases using PROTACs?
So there are a number of targets that are involved in the immune response that you can imagine you would want to tamp down if you have an overreactive immune system and that's targeting self-antigens. And so, yes, I think that it is conceivable. And there are other companies that are focusing on additional non-oncology targets. And so I think that they're either in the clinic or soon will be in the clinic with something that could be applied for autoimmune.
Thank you. If anybody has any questions. Thank you very much for an amazing talk. I have found it really difficult to imagine that 25,000 PROTACs can be developed and targeted also. And I hope that it would be possible to solve the problem of many diseases, autoimmune, pathogen-based, and sudden diseases like COVID, which have never been thought of, can be treated using PROTACs one day.
I hope so too. Thank you again for this opportunity, and I wish you all the best.
Thank you. Thank you very much.
Bye-bye.
Yeah, bye-bye. So we come to the end of the session today, and we'll be starting at 9 tomorrow morning. We would have Christine Tooley's lecture in the morning. Because of the time zones, we have to start very early so that by 11:30 or so, she would be able to complete her talk. And then we would be 11:30 in the US. And we would continue with rest of the presentations of the day. So we meet tomorrow at 9 a.m.
Yes, ma'am. Bye.
Bye-bye.
Bye-bye, everyone.